Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PUC first and second year. If you like my channel, please subscribe, like and share and hit the bell icon so that you can get notifications as soon as videos are getting uploaded. Okay guys, we shall start today's video. In today's video we shall discuss about oogenesis. In our previous video we have discussed about mammary gland. Today we shall discuss about oogenesis. In your textbook, in NCERT textbook, you'll find on page number 49, and this is the schematic representation of oogenesis, and its explanation you will find in the previous page, that is page number 48, which starts from here. It starts from here and completes till here in the next page. Now this structure which it has been shown, it is a section view of ovary. That is transverse section of ovary which we have shown earlier in a female reproductive system. When you take a section of it, it appears something like this. Now in this you will find these are the blood vessels, and there is a presence of primary follicle, secondary follicle, tertiary follicle, Graafian follicle and a ova. Then comes the corpus luteum. Now what are all this? When I shall discuss about complete oogenesis, then you will understand. First I shall discuss about the theoretical explanation of it, then we shall discuss about its schematic representation. So as we have here, that is oogenesis. In this oogenesis you will find the process of formation of mature female gamete. The process of formation of mature female gamete is called as oogenesis. Now that mature female gamete can be ovum or a ova or an egg. These are the different terms, different words which we are using for mature female gamete. The process of formation of mature female gamete is called as oogenesis, and it is similar to spermatogenesis. As we said, the process of formation of sperm is called as spermatogenesis. But when we discuss oogenesis, oogenesis is completely different from spermatogenesis. How it is different? We shall see here. Here in oogenesis, oogenesis starts in an embryonic stage. Which embryonic stage? When the female is in the form of embryo. After fertilization, when does the child is when an embryo has been formed, if that embryo is going to develop into a female, in that embryonic stage itself the oogenesis begins. Whereas this point we have never said in spermatogenesis, because spermatogenesis can take place throughout the life of the male, whereas in females oogenesis starts from the embryonic stage. In an embryonic stage, millions of gamete mother cells are formed. That millions of gamete mother cells are called as oogonia. Those millions of gamete mother cells are called as oogonia, and that oogonia will be diploid, right? The oogonia is what? It is a gamete mother cell. As you know, gamete mother cells are what? They are called as meiocytes. Meiocytes are diploid, as you know very well. Now these oogonia, these are formed where? In the fetus. Fetus is nothing but the embryo. Embryo when it is going to develop like a female, embryo when it is going to develop like an human being. Here when it is going to develop, we call it as fetus, and in the fetus ovary oogonia are formed. How many oogonia? There are millions of oogonia are formed, and these oogonia are diploid. Now no more oogonia are formed after birth. After birth of the baby, of the female baby, no more oogonia will be added. What oogonia will be added? In an embryonic stage, same number of oogonia will be continued, but no more oogonia can be added after birth. Now oogonia will start its division. Which division? It will start its meiosis 1. It will start its meiosis 1 and started with the prophase 1. As you remember, meiosis 1, it has prophase 1, metaphase 1, anaphase 1 and telophase 1. Here oogonia will start its division with the prophase 1 of meiosis 1, and it will temporarily get arrested, which means it will temporarily get stopped. Where? After prophase 1. It is not getting continued with the metaphase 1 here. It is getting arrested at a primary oocyte stage. Now oogonia, it has started division prophase 1 and it is got arrested, which means it has been stopped, and that division has been stopped at a primary oocytes. Now what stage is from primary oocyte? Now prophase 1 means it is not getting reduced to the number of chromosomes as haploid. The number of chromosomes will remain same, so primary oocytes will be diploid. Now these primary oocytes, those are surrounded by layer of granulosa cells. Primary oocytes, those are surrounded by layer of granulosa cells. If I will show you here something like this, these are the oogonia. Oogonia, first it will divide by mitosis to increase the number. How many numbers? That is in millions, as we have said earlier. Now after this Oogonia have been formed, they have started with the meiosis 1 and they have been arrested with that stage, and that we call it as primary oocyte. That primary oocyte is diploid, right? Now these primary oocyte, that's primary oocyte, if I am showing it here, okay, if this is, we shall say this one as Oogonia. These Oogonia will divide by mitosis, increase in number. I will show only one here. Now this we are calling it as primary oocyte. Now this primary oocyte which we have shown, this one is, first it has been started with the mitosis and it has been differentiated. Now only prophase 1 has been completed. Okay, after prophase 1 it has been arrested. Now Oogonia, we said it is diploid, then primary oocyte will also be diploid, right? Now this primary oocyte, now it is getting surrounded by layer of granulosa cells. This is a primary oocyte. Now it will get surrounded by layer of granulosa cells. Okay, now this primary oocyte, when it is having a layer of granulosa cells, now for this we call it as primary follicle. Okay, when it is having this granulosa cells, we call it as primary follicle. Now this primary follicle, if you find primary follicle, there are large number of these follicles which degenerate from birth to puberty. Okay, now primary follicle has been formed. For primary oocyte, what we said, the primary oocytes, those are getting surrounded by layer of granulosa cells. For those we call it as primary follicle, and these primary follicles are large in numbers, and large number of these follicles will degenerate when from birth to puberty. Puberty is nothing but when a human being is getting mature. For that we call it as puberty. Okay, large number of these follicles, which follicle? Primary follicle, will degenerate from birth to puberty. How many they will be left? Nearly around 60,000 to 80,000 will be left in each ovary. In each ovary, 60 to 80,000 of primary follicles will be left. Okay, earlier how many those were? In millions. As we have said here, those are in millions, right? We have said in millions. Now what happened after birth? What primary follicles have been there? Those have been started to degenerate. Till when? Till puberty. And till puberty, when they are reaching, they are in this number. Okay, now this primary follicle was what? It was a primary oocyte which was surrounded by granulosa cells. Now this primary follicle, it is surrounded by more layer of granulosa cells, which means now this was a primary follicle which we have shown. If I draw it separately, it will be something like this. Just roughly I am showing. Okay, this is the primary follicle. If I am showing it like this is a primary follicle. Now this primary follicle will be surrounded by more layers of, okay, will be surrounded by more layer of granulosa cells. When more layer of granulosa cells it has been surrounded, we call it as secondary follicle. Okay, we call it as secondary follicle. Now here secondary follicle is what? As we have shown here, it is surrounded by more layer of granulosa cells and new theca. It is called new theca, is nothing but theca is covering, new layer. Okay, and for that we call it as secondary follicle. Now this secondary follicle will soon transforms into tertiary follicle. Okay, now this was secondary follicle. This secondary follicle will transform into tertiary follicle. Now in tertiary follicle there are two different layers. Okay, in that it consists of two theca, a theca externa and theca interna. If you find here, so tertiary follicle, it is having fluid filled cavity called as antrum. How to identify tertiary follicle is, it do have fluid filled cavity called as antrum, and it has a theca. Theca has been said layer or a covering, theca interna and a theca externa. So this one is, outer one is theca externa and the inner one is theca interna. Okay, these are the two layers outer side of it, and it do have a presence of a fluid filled cavity. Now this here center one, this one is, which I have shaded, that one is antrum. It is a fluid filled cavity. Okay, antrum is a fluid filled cavity. This cavity is found in a tertiary follicle, not in a primary nor in a secondary follicle. It is directly found in a tertiary follicle. Now this structure we call it as tertiary follicle, right? Okay, if you found here tertiary follicle, it is fluid filled cavity with the antrum. theca, it has theca interna and a theca externa. Now in this tertiary follicle there will be presence of primary oocyte. Okay, these are the outer coverings which we are discussing, right? Primary oocyte was there. Primary oocyte, it was covered with one layer of follicle, we call it as primary follicle, and one layer of granulosa cells we call it as primary follicle. Many layer of granulosa cells we call it as secondary follicle. Here it has started to transform that with a fluid filled cavity and a theca externa and interna, tertiary follicle. Now inner side of that, what it is present, it will be remaining same, that is primary oocyte. Primary oocyte grows and completes its meiosis. Where that primary oocyte will be present? Here, this one, center one, as we have said, right? This was the primary oocyte, it was covered by the granulosa cells. So similarly here also, if I am showing, I will show something like this, and here I will show like this. Now this one is what? Primary oocyte. Okay, this one is primary oocyte which we have shown here. This primary oocyte was covered by many layers, okay, which is present here now. Now what happened to that primary oocyte? Now this primary oocytes, if you find here, this is the primary oocyte. Okay, primary oocyte grows and it completes meiosis 1. The primary oocytes which has started with the meiosis 1, prophase 1, it was arrested. After forming the tertiary follicle, then it completes its meiosis 1, and then it forms a secondary oocyte and a first polar body. Okay, secondary oocyte and a first polar body will be formed, and this secondary oocyte will be large in size, and it is filled with a nutrient rich cytoplasm. Large amount of nutrient which is rich in cytoplasm, that will be present in a secondary oocyte. Okay, so I hope you understood till here. Then okay, now secondary oocyte is formed and a first polar body. If I show you here, now this is the tertiary follicle we said. Now it do have presence of primary oocyte in that. Now this primary oocyte will divide, it will complete its meiosis 1. Completes meiosis 1. How it completes meiosis 1? With a metaphase 1, anaphase 1 and telophase 1 will be completed here. Now when it has been completed, it will result into 2 cells. So which 2 cells? Something like this. Okay, this one is secondary oocyte. Okay, this one is secondary oocyte, and this one is first polar body. The smaller one we call it as polar body. Okay, first polar body, right? Then you can find further, we shall discuss here. Now tertiary follicle, this tertiary follicle which has theca externa interna with the intron, that tertiary follicle will further change into mature Graafian follicle. Okay, when it has formed secondary oocyte, then the tertiary follicle, the outer coverings will change into mature Graafian follicle. Now this mature Graafian follicle, okay, it do have secondary oocyte in it and forms a new membrane called as zona pellucida, right? Okay, now this secondary oocyte is present. Here we say it as mature Graafian follicle, bit large size and a fluid filled cavity is present in drum. Okay, similar to this structure you will find. Now this secondary oocyte will be having Graafian follicle. Now it is mature follicle or a Graafian follicle. This was tertiary follicle. Now it is tertiary follicle will be transformed into mature Graafian follicle. Now this mature Graafian follicle, it is having an outer covering, one more layer of covering membrane called as zona pellucida. This zona pellucida we have used in a structure of sperm. There are some enzymes which are present in the acrosome. That enzymes are responsible for breaking down or rupturing this layer called as zona pellucida. There we have discussed. Okay, now secondary oocyte is formed. Then if we continue, now the secondary oocyte forms a new membrane called as zona pellucida. Now secondary oocyte, it is having Graafian follicle will rupture. Later Graafian follicle will get ruptured and release secondary oocyte. Okay, so what Graafian follicle we have said? Now this we said it as Graafian follicle. This Graafian follicle will get ruptured and it will release this secondary oocyte outside. Okay, for that process we call it as ovulation, right? For that process we call it as ovulation, right? So this ovulation is nothing but the process of release of ovum from the mature Graafian follicle. We call it as ovulation, but actually ovum is not released, that is secondary oocyte is released. Okay, Now this secondary oocyte will be released where I shall show you, right? Okay, coming back to the diagram. Now this is the structure of the ovary, right? TS of the ovary. Now here you can find there are presence of primary follicles. If you find your in your textbook, it is very clear in that. So there is presence of primary follicle. Now primary follicle is what? It is having primary oocyte inside it. Now it is having only one layer of granulosa cells. Now this primary follicle is getting converted into secondary follicle. If you find carefully, then the number of granulosa layer has been increased. Again it is having primary oocyte. Now it is getting converted into tertiary follicle. Okay, tertiary follicle is here where you can find fluid filled cavity. Here fluid filled cavity is the yellowish in color. That one is nothing but elytrum. Now this tertiary follicle, what does it consist of? It consists of secondary oocyte. Why? Because it has completed, now primary oocyte, it has completed its first meiosis, resulting in the formation of the secondary oocyte. And this tertiary follicle is getting transformed into Graafian follicle, mature Graafian follicle. Here you can find secondary oocyte will be clearly visible. Okay, and then later it will form an ova. Okay, now this mature Graafian follicle will get ruptured. Because it has been ruptured, that ova has been released out. The process of release of ova from a mature Graafian follicle, we call it as ovulation. Now ovum has been released. Now what has been left? Only the outer covering. That outer covering is nothing but that outer covering is nothing but the layers which are present on the Graafian follicle. Which layers? That is theca externa, theca interna with the granulosa cells. That will be left over. Now for that we call it as corpus luteum. Okay, if you can imagine like a balloon, inside the balloon if air has been removed, how it appears. Similarly, here Graafian follicle will get transformed into corpus luteum. Now this corpus luteum will be retained in pregnancy if fertilization takes place. If not, it will get degenerate. Okay, corpus luteum and corpus albicans, later it will get degenerate. Now ovum which has been released here, that is caged or that is held by fimbriae, that is fallopian tube. Okay, now if you go through this structure, coming back to the schematic representation, in this structure first what they have shown, it is oogonia which is diploid. It is undergoing mitosis. It has been increased in the number and it has formed primary oocyte. This one is the primary oocyte. Primary oocyte is diploid. In this they have not shown about it. Primary oocyte, it is having one layer of granulosa cells, then two layers of granulosa cells forming primary follicle, secondary follicle, tertiary follicle. That has not been shown. So if you see directly, primary oocyte is formed, that primary oocyte is diploid. First meiotic division completes, forming secondary oocyte. Okay, now this primary oocyte, it grows. Okay, it undergoes a growth phase. It grows with a nutrient to the cytoplasm, and then it completes its first meiosis to form one large size, one small size. Large size we call it as secondary oocyte, small size we call it as first polar body. Now this secondary oocyte, again it will undergo meiosis 2. After meiosis 2, it forms again one large size called as ovum, one small size we call it as second polar body. Okay, this is the simple structure to draw for schematic representation. For explanation, you have lot of information as I discussed earlier. Now, if I say oogonia to primary oocyte, what division? Mitosis. In that, it has also started with meiosis 1, prophase 1, and it has been stopped. When it is getting converted to primary oocyte to secondary oocyte, it has been completed meiosis 1. It has undergone changes from primary follicle, secondary follicle, and tertiary follicle. All those changes have been taken place here. Later, meiosis 2 takes place in this and then results in a formation of ovum. Now, question is what happens to this first and second polar body? Still, it is till now we don't know what is the actual function of it, but it has been produced. Later, it will get degenerate. Now, here you will find that fetal life they have shown. This is the part which takes place in the fetus stage, that is in an embryonic stage, that is oogonia to primary oocyte. What changes takes place? That will take place in a fetus stage or in an embryonic stage. Now this stage what they have shown, birth, childhood and puberty. Till in these three phases you will find that there is a growth phase. Then comes adult reproductive life. In an adult life, when an individual has been mature, after that you will find this primary oocyte will form secondary oocyte, then secondary oocyte will form an ovum. This is the schematic representation. If they ask write the schematic representation of an oogenesis, you are going to draw similar to this. Okay, same this diagram. If they ask to explain, while explaining you will talk about primary follicle, secondary follicle and tertiary follicle. All those will be explained in that. Right? Hope you understood this. Okay, Now, function of corpus luteum, it is nothing but it secretes one of the hormone called as progesterone, which is helpful for maintaining pregnancy and releasing for easy delivery of the baby. Okay, corpus luteum is going to secrete one of the hormone called as progesterone, right? Hope you understood this. And one more point I would like to add. This second meiosis actually when does it takes place? Second meiosis takes place when this secondary oocyte it has been released, as if you find in this diagram. This is the structure of ovary. Actually, when a Graafian follicle will release, it is not an ovum, it will be a secondary oocyte. That secondary oocyte will be released here and it will be cached by this fimbriae. It will hold that egg and it will release that one into this fallopian tube. Now, when it has been reached here, when sperm will come and get fertilized, when sperm will start to penetrate, after that second meiosis will complete in a secondary oocyte and it will produce an ovum. Then fertilization takes place. This is the actual case. When I am discussing about fertilization, at that time I shall discuss about these two. Okay. So this is how ovary, how it will release ova and cast by the fimbriae and enter into the fallopian tube. Now fertilization takes place in the ampulla isthmic region of the fallopian tube. Okay. So this is about the your oogenesis, very important as important spermatogenesis. So they have given the number of chromosomes, diploid number and a haploid number. From secondary oocyte it will be haploid, in primary oocyte it will be diploid, and oogonia will be diploid. Okay, hope you got this, you have understood the concept. If you have any queries, any doubts, please let me know, comment it in the comment box, and I shall meet in my next video with new ideas, with a new topic that is menstrual cycle. We shall meet soon. Stay safe, stay healthy.